overview of the transamination reactions, as well as going over biosynthesis and the things that you did already have on your test. But we're going to split it between the two because there's actually, in the pathway of the urea and transamination synthesis, I want to hit that in more. And so what I have today is going to be kind of a preview rather than every detail. So before we go into it, are there any questions that we want to start with? Nope. Okay. Excellent. Then the idea of protein metabolism. How are we going to handle excess protein? So when it comes to carbohydrates, when we have extra of that, we go ahead and we turn it into excess protein. So when it comes to carbohydrates, when we have extra of that, we go ahead and we turn it into either fat, or we turn it into glycogen, and we turn it into energy and store it. And we can have a storage form of that glycogen, etc. Fats, we can store them in fat as well. Protein, we don't really have a source of protein that is stored as itself. And so whenever we have extra protein, we're not going to build extra muscle because muscle has an upkeep cost. And so if we just built more muscle to store the amino acids, then that would actually be increasing our baseline calorie need. It would make it very hard for hunter-gatherers and things like that to continue to get as much food as they need. So what we do is we actually break down excess amino acids pretty quickly rather than storing them. So I'm going to go ahead and draw a muscle here. This is my form of a muscle. And that's one place that you can have this. So if you're in starvation, you can pull them out of your muscle. Or it's just any excess amino acids that you've eaten. Either of these sources are going to go through transamination. Okay? So amino acids, they have, depending on which textbook you read, there's at least 21. Some textbooks throw in some subgroups, but there are lots of different kinds of amino acids. So in the muscle, we're going to take all of these amino acids and we're going to use what's called transamination. And let's do a little bit of vocabulary before we go further. Amino acid, it has carboxyl on one side, hydrogen on another, and then an amine group and an R group. R group just means anything can go here. And then the amine group has a nitrogen in it, NH2 or NH3. So this is what we call an amino acid because it has an amino group, a nitrogen group. If we take the nitrogen group off, and we just stick another hydrogen there, say, then the name changes into either ammonia or ammonium, which is going to be the nitrogen that's been taken off. And what's left behind has no differences except the lack of a nitrogen group, and this is going to be called either a carbon skeleton, or more correctly, an alpha keto skeleton.
And the COO is, you said it was carboxyl group?
Yeah.
Okay.
And so when we're talking about going from an amino acid to an alpha, another name for this is alpha keto acid as well. Alpha keto acid. To an alpha keto acid, we're literally just saying we popped a nitrogen off. Now there are a few amino acids, 2 of them, where you can have 2 nitrogens being part of the amino acid. And so we're going to go ahead, COOH, and they have two amino groups that were part of them, and you were able to pop both off. And so an alpha keto acid means that it is one nitrogen less than what it started as. And so if you have an amino acid that started with two nitrogens and you take one off, it still has one left. That would, even with one left, it can be called an alpha keto acid. Because an alpha keto acid just means minus one nitrogen. Okay? And so, let me give you an example. Glutamine. Glutamine has two nitrogens. If you take one of those away, it becomes glutamate. Because glutamate looks exactly like glutamine, except has one less nitrogen. So you pop one off, and ta-da, you have glutamine. And I'll go ahead and write like that. If you pop one more nitrogen off of the glutamate, you're going to end up with something called, it should be familiar, alpha keto glutarate. You guys have seen that. So times 0. And there's no nitrogens. There's just nothing. So question for you guys. I'll answer one of the things. Which is a carbon skeleton? This is going to be a carbon skeleton because it's got no nitrogens. If we started with glutamine, which of these two would be glutamine's alpha keto acid? Remembering that it means minus one.
Glutamate?
Yes.
Okay.
So the alpha keto acid is just minus one nitrogen. And minus one from glutamine? Glutamate. Minus one from glutamate? Alpha keto glutamate. So our carbon skeleton means there is only carbon left, no nitrogens. Alpha keto, let's get rid of alpha keto skeleton in the middle there, it's kind of a useless term. Alpha keto acid just means one less nitrogen than the other guy that we were talking about, the original amino acid.
So you said glutamate was the
Alpha keto acid of glutamine. Whenever you say alpha keto acid, try to say of and whatever the original was. That way you won't get confused. There should always be an of, because alpha keto acid is always part of a pair. So, going back to the metabolism, when we're trying to break down amino acids for energy, we are trying to take this semi-toxic. Nitrogens are very toxic when they're free-floating. And so we're trying to take this toxic nitrogen and be very careful with it so that it gets taken care of and won't damage anything. And eventually we turned into urea and then peed out. The carbon skeleton with no amino acids, that will then just be burned for energy. And alpha ketoglutarate, where have you seen that before?
on the TCA cycle?
Yes. This is one of the intermediates of the TCA cycle. So as soon as you pop off all the nitrogens for this particular amino acid, it's already in the middle of the TCA cycle, which is basically the burn me cycle. It's already ready to be burned for energy without any further modification. Now,
Sorry, did you say what happened to the nitrogens?
we haven't gotten there yet. No, I
I
didn't.
want to make sure I didn't miss it.
So eventually, I guess I did sort of say, eventually they'll be turned into urea, but I didn't go any deeper than that. That requires drawing, so we'll get there.
So which one goes to urea?
Nitrogens will go to urea eventually. All amino acids nitrogens will end up going to urea. Whether it's threonine or glutamine or alanine or aspartic acid, doesn't matter. Any of those, they all have nitrogen, pop them off. Now, when we take the nitrogen off, I drew it here as ammonia, which is what it is when it's free floating, and usually free floating is not good because that's toxic. So usually we're going to stick the nitrogen onto something else, and if we're taking the nitrogen from one thing and putting it on to something else, we aren't really deaminating something so much as we are transaminating something. I'm just going to draw a random molecule for now, and if we stick the nitrogen on there, it never got free, right? And so this would be an example of transamination. If we took the nitrogen and just set it free, which we do in some rare occasions and sometimes when we don't want to, that would be called deamination. So trans is like transferring it, D is like taking it off and throwing it free. Good so far? All right, progressing then. With this, I'm going to go ahead and draw a little bit more of what we could transaminate to. But first, actually, before I do that, I want to explain why it might be something we want to do. So if we took all the amino acids from the muscle and that we ate wherever, and we throw them into the bloodstream, then all these amino acids, there's 21 different kinds. That means that the liver, who's going to be, of course, the star player later on, because that's always the star player, the liver is going to have 21 different amino acids that it's looking for, and it's going to have to be able to grab 21 of them effectively, and it's going to have to blah, 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 21, and it just makes it really hard, and it doesn't know, like it has to have different transporters for everything, unless it does something different, which it does. So this is not what happens. This is the more complicated way that thankfully we don't do it. We actually are going to take all the amino acids in the muscle, and we're going to transport them, transaminate them, rather, from 21 types down to 1 type. So we just convert all these 21 into a single amino acid, and then that one kind, we send a frick ton of it out into the blood, and then the liver only has to be able to be really good at picking up one kind, and it watches for a lot of that one kind, and it scoops it up. And so then it can be very specific, and it's much easier to regulate because you only have to regulate one transporter on either end. You only have to regulate one breakdown cycle on either end, not 21. So
Why would it need to regulate? Why wouldn't it just, whenever it came by, just metabolize it?
good question. Why does it need to regulate? Why doesn't it just always grab it? Alanine is going to be the star player, and sometimes some cells need alanine. Or if 21 were coming out, cells need all the different amino acids. And so if the liver was always grabbing all of them, then the rest of the body wouldn't get any. So if the liver grabs too much, then the rest of the body suffers. So the liver needs to only grab it when there's a ton in the bloodstream or when there's more than normal. And it's like similar to how the liver picks up glucose. The liver is weaker at picking up glucose. That way everyone else has a chance. The liver is going to be good at picking up alanine only when there's a lot of it, just like it's only good at picking up glucose when there's a lot of it. So it's always the one that's waiting until there's a ton of it, and then it will start being better and better at grabbing it. Did that answer your question? Okay, good question, by the way. So we're going to go ahead, and this is one of the, we're going to go ahead and talk about transamination. This is one of the transaminations that you should be familiar with. The sequence is glutamine to glutamate to alpha ketoglutarate. You can also go the other direction. Alpha keto to mate to amine. The other amino acids sequence that you want to be familiar with is alanine. And then we'll do one more also. Alanine goes down to pyruvate. So alanine has one amino acid. Pyruvate has 0. Nitrogen group, I should say, whatever. It has one amino acid itself. Good job. Amino acid's the whole thing. Amine group. So there's another pair. What happens then if the muscle has a bunch of pyruvate and it takes the other 20 amino acids and has them throw all their nitrogens at pyruvate? What would pyruvate become if you added a nitrogen to it? Sorry?
So then would it be alanine so it can go into the bloodstream? Is that?
Exactly. So by adding a nitrogen to the pyruvate, it becomes alanine. Alanine is then, all 20 of them have been turned into alanine. Alanine goes into the bloodstream and will be accepted by the liver in particular, as well as anyone else who needs alanine, but the liver is going to be the one that we're getting into. So, what happens then to these 20 other amino acids? These 20 other amino acids, once they have no nitrogens, they're no longer amino acids, so now they are all carbon skeletons. And carbon skeletons, we know two carbon skeletons already. One of them is alpha ketoglutarate, one of them is pyruvate, and I'm not going to throw all the others at you, but you'll notice that both of these are really close to the TCA cycle. Pyruvate has like 2 steps away from the TCA cycle. Alpha ketoglutarate is actually in the TCA cycle. All the rest of the carbon skeletons have a way to get to the TCA cycle. Lots of them are actually part of it. And so once they have skeletons, all of them will end up in the TCA cycle. And if you burn something in the TCA cycle, what do you get?
Energy.
Energy. So you'll get energy. So, as a recap, the muscle spent a lot of pyruvate to make a lot of alanine by stealing all the aminos off of the amino acids. So then you have a bunch of alanine, and you spent a bunch of pyruvate to make it. That means that the muscle just lost a lot of energy because pyruvate was going to be energy. But all the carbon skeletons kind of replace that lost pyruvate energy because then you just burn them instead. So the muscle ends up being roughly energy neutral. It didn't lose anything. Does that make sense? It spent this to make alanine, but then all the leftovers, it burned those instead, still got energy.
And the 20 is yielding the nitrogen that's adding as the precursor to alanine for pyruvate?
Right, so the 20 other non-alanine amino acids, they gave away their nitrogen, and then what was left of them, the leftovers, the carbon skeletons,
For energy.
came down and burned for energy, exactly. The enzyme that takes the nitrogens and puts them onto alanine is alanine transaminase, because that's what it's doing. It's transaminating into alanine. You'll frequently see this shortened into ALA. Same thing.
the other way, can alanine degrade into pyruvate for
Mhm.
some reason if it needs to?
It definitely can, and that is what will happen in the liver. You will have alanine in the liver once it's there. Alanine is going to give away its nitrogen. And it can do it in a couple different ways. Most often in the liver is going to go ahead and make urea. And it's like, but that's not the kidney. How's it going to get to the kidney? I'm reading your mind. So and the whole urea cycle, we'll see if we hit that today or if we hold off. Basically, once you have the nitrogen, then you just go through a series of steps, and at the end of it, it's urea. And it just all happens in the liver. Urea, however, is much less toxic than ammonia. So free nitrogen, really bad. And we'll talk about why in just a second. Urea, it is toxic because it has a lot of nitrogen, but it's much less toxic because it's bound up and it's not as, we'll talk about blood-brain barrier and stuff like that in a little bit. But the urea, to answer your question, is going to come out, and we can actually handle, and this might be something that she wants to go over again or have you remember, we can handle urea in the blood. The normal human has urea in their blood a little bit all the time. And when it's in the blood, what does all blood eventually pass through? There's a lot of correct answers to that, but the one that I'm thinking of is kidneys. So the kidneys are going to have that blood go through, and it's going to filter the urea out. All right, I'm going to get rid of all these words here that I didn't.
Why does the pyruvate have a 0 on the end?
Because it had no nitrogens.
Okay. And alanine has 1.
Right.
So would you say that pyruvate is the alpha keto something alanine?
Yes. Alpha keto acid of alanine is pyruvate. Exactly. All right. So if urea is made in the liver, and there's a big pathway to that we'll hit in a little bit, but the concepts are what I want to hit first. Because honestly, the hardest part of this is getting it all straight. And then once you have the foundation, you can do the rest of it not too bad. Because the urea cycle is like 1 page. It's actually, ta-da, you just type it in.
Well, and then her slides don't always have that overview. Her slides are a lot of details usually. So.
That can happen. Thankfully, the urea, she actually does have a really good slide for the urea cycle. I actually saved that slide. It's like really good. This part I didn't find a single slide for, but all right. So urea travels through the blood to.
We got our one sheet now.
To the kidney, and the kidney will filter it out, and renal pelvis goes down the ureter, and eventually becomes urine instead of just urea.
What mixes with it? Just water from the kidneys or other things?
You guys will have kidney physiology on your favorite day, whatever day that is. It will become your favorite day because kidneys are fun. Basically, yes, there will be some water, and then you filter out a bunch of other stuff, and then some of it's like, no, wait, I want that back, and so you pull it back, and then how do we do that is a bunch of little things. But simple answer to what else goes out with it, a little bit of water, some other wastes, and then a few things that are semi-waste, semi-not, and it's like sodium, we need that to go out so that water goes out. So in the kidney, we have urea. And I'm going to put that there with the number 1 because there's going to be a second thing that goes to the kidney as well. Because the kidney can also get nitrogen prepped up with our second story here. But first, right here, alanine to urea, that's going to be the beginning of the urea cycle. So so this one right here, alanine is not actually transferring it to a molecule called urea because urea hasn't been made yet. Urea is actually made from scratch from free nitrogen, which is basically ammonia or ammonium. And I drew it in purple instead of green. Sacrilege. So alanine actually lets go of its nitrogen without putting it on anything else. So it's not transamination in the liver, it's deamination. So right here we have deaminase. Now, with that free ammonia, this is where stuff can get a little janky if we don't make urea. If your liver is damaged and it can't make urea as quickly as the ammonia has shown up, then we get to see why is ammonia bad. If you have enough ammonia build up in the liver, some of it's going to start escaping into the bloodstream. Bad thing about ammonia is that it can cross the blood-brain barrier, and it will, and it can get to a lot of other places too, but they're not as sensitive as the brain. So other places are suffering while we talk about this, but not like, it's not what we're concerned about. In the brain, however, if we have ammonia, we talked about alpha-ketoglutarate being in the TCA cycle. What happens if alpha-ketoglutarate gets an ammonia? Glutamate.
You get glutarate is up there or glutamate?
Glutamate. So without wanting to, the TCA cycle, so every brain cell is going to be running the TCA cycle because it needs energy too. Every cell, except for the red blood cells, run the TCA cycle, right? It's not just the liver. So the TCA cycle that's going on in the brain, TCA, got this little wheel making energy so that the brain can chew up sugar and keep going. Without that, you can't chew up sugar, can't keep going. So the TCA cycle is not just the liver, it's everywhere. So the brain's depending on that to make brain's ATP, right? So if we make alpha-ketoglutarate and we take it out of the TCA cycle and turn it into glutamate, what happens to the TCA cycle, do you think?
It doesn't go function because it's missing.
Right?
Yeah.
It doesn't go anymore because it's missing one of its pieces. So every time it gets some sugar and it makes it into acetyl-CoA and it cranks it through a few steps and it turns into alpha-ketoglutarate, then right there, nitrogen grabs it, turns it into glutamate, and it's gone. And it doesn't get to the energy-producing steps. So your brain could have a frick ton of sugar, but if there's enough nitrogen there that it's always bumping into the alpha-ketoglutarate randomly and stealing it away, you get 0 sugar. Yay!
0 ATP.
0 ATP. And then the brain doesn't have any energy, doesn't have any ATP, can't run any processes, and it starts to shut down. But wait, there's more. So besides your brain just starving to death, glutamate. If we add a nitrogen to glutamate, what happens to it? Glutamine. Fun little thing, nitrogen can cross the blood-brain barrier. Glutamate can't. Glutamine really can't because it's got a bunch of other stuff attached to it, and so it can't just float through. It's not hydrophobic. It's too big for one thing, and then it's also, membranes won't let it through. So glutamine also, it's got a bunch of charges because of those amino groups. And have you guys ever heard where salt goes, water follows? Well, salt has a charge. That's why water follows it. This is basically really, really salty. If you want to think about it that way, it's very charged. So then you have a lot of water come out of the blood through the blood-brain barrier and goes to the glutamine. The glutamine, if it could float back out into the bloodstream, okay, well, brain's still starving to death, but hey, water would be equal on both sides. But the glutamine can't leave, so the water has to come to it.
This is so exciting.
Yeah, right? Someone else has a macabre sense of humor. All right, so the brain starts to get more water coming in around the glutamine, and that continues to happen until the brain gets so big that it actually, you guys know the foramen magnum at the base of the skull? Brain gets big enough, it actually swells, and then it forces the back part of the brain through the foramen magnum, and it's called a cerebral hemorrhage. Hernia. And so your brain herniates, and that is usually what causes death. You don't have time for your brain to starve to death. Your brain gets squished through a small hole. Yeah, so ammonia is toxic.
What's the name of that, again?
Cerebral hemorrhage. I said hemorrhage. Hernia. Thank you. I do that like every time. So the glutamine pulls water in, which leads to cerebral hernia. So it is both starving to death and herniating.
And nitrogen is just available to just be a catalyst to anything that's outside at this point.
Nitrogen can get anywhere because membranes don't stop it, blood-brain barrier doesn't stop it. It's just it's small enough and it's hydrophilic and hydrophobic enough that it can permeate membranes, and so it can go through. As soon as it attaches to the glutamate, the alpha-ketoglutarate to make the glutamate, and also true for the glutamine, that's too big to just float through the membrane, so then it stays there and it builds up, water follows it, and then it expands. So
why would there be a buildup of ammonia? What would be a common cause for that?
Common, thankfully it's not common, but what would be one of the more common causes that we have to pick from would be if this enzyme, deaminase, if that was broken, then that would be bad. You wouldn't notice. There actually are people who have, not deaminase. Deaminase would just mean that you'd build up alanine. You wouldn't build up ammonia. In the urea cycle, let's actually go into the making of urea a little bit. I'm going to do that down here. So we're going to have this zoomed out right here. Here's the urea cycle. This side is inside the mitochondria. This is outside the mitochondria. So there we go. All right, so first step was, there are two steps, and there's one enzyme that grabs the ammonia and it puts it with a carbon dioxide, and then there's a second step that takes that and turns it into carbamoyl phosphate. So it's going to be carbamoyl phosphate synthetase. Carbamoyl phosphate synthetase 1. That's one enzyme. And then there's another one whose name I don't remember. So if you guys have the slides, that would be great. But if not, this first guy, if he is broken, he can't grab the ammonia, and you have a lot of ammonia build up quick because you can't even start the process. If the first guy works, then he can grab the ammonia and turn it into this middle molecule whose name is going to be a mysterious surprise for when the slide comes. I prepped for a different lecture, so this is... the nitrogen can then be attached to this molecule, and that's much less toxic. And so you can build up a lot of these nitrogen-linked molecules, And that's better than free ammonia. And so, but if carbamoyl phosphate synthetase is broken, you can't take these guys and turn them into the next thing. And so you just keep building up all these nitrogen-linked molecules until you have so many of them that the reaction starts going backwards. And then you get the ammonia back again. Because if you build up a lot of products, it'll go from products to reactants. Because that's just what happens with chemical pathways. If you build up a lot of a product, most of the time, it'll then start going backwards. So if you have the first guy broken, then breaking down any protein, you would someone with that disease would be trying to avoid eating protein except for exactly how much they needed. They would never want to eat any extra protein. Any extra protein would then they'd try to turn it into ammonia, urea, and they couldn't, and then they would have this starting to happen up here.
Is there a way to decrease the brain swelling? Like, can you drain it or?
You would have to be whoosh. Yeah. If you were already in the ER, maybe. It's pretty fast. Usually that's not a good thing. It's never a good thing. But I don't know of anything that you can really, let's see, what could you do? Oh, you could do dialysis. I know what to do if you drink. There's another way that you can have brain herniation where you drink a frick ton of water, and then you have so much water that the osmolarity changes, but that's not related to ammonia. But then what you do is let's see, what did you do for that one? I think you either gave him a bunch of salt or oh well, I don't remember. Okay. Don't have it happen.
There's not a cure, that's what I'm hearing.
Not one that I know of. So dialysis would help, but I don't know if it'd be fast enough. So if the first enzyme is broken, then you're just gonna have to avoid protein really, really hardcore. If your second enzyme is broken, carbamoyl phosphate synthetase 1, then you have this kind of a buffer zone where you can have some extra ammonia and it'll be turned into these little intermediate squiggles. And those aren't toxic, but you can only have so many of them before they start changing back. So first one, really rough. Second one, you got some wiggle room. Okay? Does that make sense? All right.
And the squiggly lines with the ends, that's urea, right?
That's the first step to making urea.
Urea will be a buildup of urea and lead back to ammonia. Is that what I'm seeing?
Not exactly. These squiggle lines with an N, I don't remember what they're called. That's why they're called squiggle line ends. So if you happen to have the slide, I would love to know what they're called, but it's not coming to me. After carbamoyl phosphate synthetase, you're going to have a bunch of other steps. Carbamoyl phosphate, which I'm just going to call CP. And you have a slide for this, but just in case anyone watches this who doesn't have the slides, I'll go ahead and go through it. But frankly, if you look up a slide of urea formation, then that'll be the most helpful. But carbamoyl phosphate is going to go ahead and link with ornithine. And then those two together will make citrulline. I'll go ahead and put it in a link. I'll download the slide and see if I can put it in a link in the video, or but it's definitely in one of your biochem slide sets. So ornithine plus carbamoyl phosphate makes citrulline. Citrulline, kind of like citrate. Citrulline can cross the mitochondrial membrane. It gets outside, and then you have arginosuccinate. Yeah. So carbamoyl phosphate plus ornithine equals citrulline. You just put them together.
Oh, citrulline, not citrate.
And the enzyme name that does that is going to be
Aminophosphate synthesis.
that was the one that made the carbamoyl phosphate.
Oh, okay.
The one that takes the phosphate and the ornithine, the carbamoyl phosphate and the ornithine, and puts them together. That's going to be trans-carbamoyl phosphate. Let's see. Trans-carbamylase. There's ornithine in there somewhere. Trans-orno-carbamylase. That doesn't sound right. Carbamoyl phosphate.
I didn't see that in our site.
It's in there. It's in one of them.
Yeah, it might be from next week. I don't know if we have access to it.
No,
Okay. Well,
we'll take it if like knowing.
We just can't help you with it.
all right. Well, I'll just power through.
Yeah.
You have transferase. That transfers these two together, citrulline, and then citrulline will be acted on by another enzyme, and that's going to make arginosuccinate is the next molecule. And after arginosuccinate, you are going to go ahead and have, arginine.
If you want to just come look at this,
Do you have it?
yeah, I feel like I Googled it, but I'm like not even able to translate it.
Ornithine transcarbamylase. Thank you. Okay, so the transferase was ornithine transcarbamylase. It's just the name of the two molecules. Ornithine transcarbamylase. So it's just naming one molecule, ornithine, and then saying that it's transferring the other molecule, trans-carbamoyl, and then ACE is on the end of every enzyme. So, and this is a bunch of details, and actually there's another video where I go into a memory thing for this. I didn't mean to go into this much detail for this, it's fine. So I'll refer you to that.
The urea cycle.
Yeah, so this is the urea cycle. And it's a cycle because arginine, when it gets acted on by the enzyme arginase, which is a really nice enzyme, it makes urea, and then it has a byproduct of ornithine. And ornithine goes back into the mitochondria. And then it can be used the next time. So really what's happening here is we made something called carbamoyl phosphate out of a bunch of nitrogen and some water and CO2. And then ornithine grabbed it, changed its name, but we don't care, grabbed it, took it all the way through, and it got swapped up a little bit along the way. It got swapped up, and then eventually arginase cuts it back off after it's been changed into urea. So ornithine is just carrying something and then whacking it against a bunch of enzymes until it looks right, and then letting it go. So all of this is just, all these names in the middle are just carbamoyl phosphate being one hammer blow closer to urea every step of the way. Then it lets it go, and ornithine comes back in to be used again. So that's what I really want you to take away from going into this, as well as the two diseases. One of them you have no buffer room, one of them you have some buffer room. With that, if you still have that slide up, they don't list it. A lot of them don't. So there's one enzyme before carbamoyl phosphate synthetase, and a lot of them don't list it, I've noticed. But if that guy's broken, then you have a big problem. Carbamoyl phosphate synthetase 1, as I'm recalling, is also the rate limiting step or the regulated step. And yeah. So hopefully, does the concept make sense? The buffer thing and the diseases, and then the fact that ornithine is just grabbing a thing and then tweaking it 40 times until it becomes urea and then letting it go. Any questions?
So when you wrote deaminase in the liver, I guess, is that only when you make NH3, or is that just like these whole processes? And that's where I'm confused.
Okay, good question. The deaminase is going to make the nitrogen, and so this nitrogen is this one right here.
Okay.
And then this blue arrow was including all of this inside of it.
Okay.
And so deaminase is where you get the ammonia, and then ammonia is what you use to build the urea.
Okay.
And so alanine, as soon as it lets go of the nitrogen, it's done with
And
its
what's
job.
that when you go from the mitochondria to the outside?
Citrulline goes through.
Citrate? No.
Citrulline. It's a new guy. Hello?
And then is the nitrogen then entering into the mitochondria to start the whole process of making urea then from the alanine after it's used by deaminase?
Good question. Question was, is the ammonia going into the mitochondria and how does it get in there and that kind of thing? Alanine actually will be deaminated inside the mitochondria.
Okay.
So it starts in there. Glad you asked that. Okay, let's see, what time are we looking at? All right, so hopefully that helps. There is one other thing that I'd like to tackle, and that is if your cells, just all the cells in the body, or you eat something that has extra protein, how the heck do they ship it off? And the liver can do this particular bit too. And it's like, they don't have ALA. Muscles and the liver are the only people who have ALA. So the cells don't have that option. What do? They turn it into a different amino acid, and the liver doesn't pick it up. Someone else picks it up. They make glutamine. So they put a bunch of nitrogens, and it makes sense that it's glutamine because then you can put two of them on because glutamine can carry two. So they just take some alpha-ketoglutarate and then pile nitrogens on there from other amino acids, just like happened over here with the pyruvate down to alanine. Now it's alpha-ketoglutarate into glutamate, I'll just put glue, into glutamine, and then you let the glutamine fly out, and the kidney, the guy you would expect to be doing a lot of this, he takes the glutamine and he uses glutamate dehydrogenase. Why am I using this? Glutamate dehydrogenase, which makes it into glutamate, and then, I think the first one wasn't dehydrogenase, this one was deaminase. Double check my enzymes, because I prepped up for what you guys were already tested on, so, but I don't want to give you that now that I realized you already tested on it. So, and then this one is going to be glutamate dehydrogenase. And both of these are going to take off one nitrogen each. So glutamine loses a nitrogen, turns into glutamate. Glutamate loses a nitrogen, turns into alpha-ketoglutarate. And those nitrogens, they're in the kidney. They're right there. So the kidney just grabs them and throws them into the urine. And so they aren't a problem.
Would you be willing to do the whole thing like really quick from we have extra proteins and then just go through it?
Gladly. All right. So we have extra proteins or we're starving. Either way, we need to break down amino acids if they're coming from the muscle. All right. Amino acids in the muscle, we're going to turn them all into alanine. To do that, we have to have a bunch of pyruvate made. And then we throw all the nitrogens onto them using alanine transaminase. That turns the pyruvate into alanine, which is then sent into the blood up to the liver. All the alpha keto acids are then burned to make up for the energy that was lost making all that pyruvate. The alanine arriving in the liver goes into the mitochondria and then is deaminated, so we take the nitrogen off of it. The nitrogen, which is now called ammonia, is going to go through the urea cycle. There's a couple diseases here. If you can't grab it, then the ammonia will get free and cause you issues in the brain, and you'll have brain starvation and brain herniation. But if it works the way it should, then you will have ornithine grab onto the carbonyl phosphate, and it will tweak it a few times, and then once it looks like urea, it will let it go. And then urea will go through the blood to the kidney. That's how you do it from muscles to urea. How do you?
Examples of other kind of cells that would do it?
Skin cells, not muscle cells, brain cells, pancreas cells, any cells.
Okay.
They will take alpha ketoglutarate, turn it into glutamate, turn it into glutamine, so that they've packed a bunch of nitrogens on there. Then once they have all the nitrogens on there, they send it out in the bloodstream. Kidney picks it up, picks the nitrogens off, drops it in the urine, picks the nitrogen off, drops it in the urine, and then the carbon skeleton that's left behind, which would be alpha ketoglutarate, it just burns for energy because it's like, it's here, why not? I'm not sending this back through the blood because who knows who would pick it up. It's mine. And that's story number 2. The enzymes of each step, go ahead and double check on your slides. But besides that, we also had vocab, which was alpha keto acid means -1 nitrogen. And if nitrogen is being put onto somebody else, we're transferring it. So it's called transamination. If we're tearing it off and just throwing it into space, it's not a transfer anymore, it's just deamination. And that's pretty much what we went over.
Quick question.
Sure.
So is this deamination when the kidney is dropping off the nitrogens into the
Yes.
urine?
Yep, these would
And-
be deaminations.
And in the first cycle, when you was going around the urea, that was transamination?
This would be deamination, and then through here, let's see. That one would be amination because you're putting it onto something. So the opposite of deamination is amination, when you're taking it from nowhere and putting it onto something. That one's much less talked about, so I kind of glazed over it. But this, you're not moving nitrogens except for this first step.
Okay.
So you're doing amination right there.
Okay.
And then all of this stuff right here, transamination. All of this, transamination. Ooh, cool tidbit. We'll end on a fun fact. So this one's clinically relevant. When we're talking about ALA and AST, ALT. Did I write ALA over here too? ALT, I always do that. Alanine transaminase, aspartate transaminase. These are enzymes. ALT, we know one place that it's found. We just talked about it today. Where do we find it? What kind of cell? Muscle. And I think I also heard someone say liver. That's correct. The liver also has it. AST, only the liver has this. And so if we see a bunch of AST in the blood, we know that there must be liver cells that are exploding, and their little bits and pieces are floating through the bloodstream, because this is one of those bits and pieces. And the only reason we'd see lots of this in the bloodstream is if lots of liver cells were exploding and sending it out into the bloodstream. That's why this is a liver health test. And so is ALT. ALT, though, if you only see ALT, it's like, is it the liver or is it the muscle that's dying? If it's the liver, you'll see both. If it's the muscle, you'll only see ALT.
You said it stands for alanine transaminase?
Yep.
And then the other one stands for?
Aspartate transaminase. Alrighty. Let's go ahead and take 5, and then we'll start back up again. I'll leave this up there for the 5. So, all right, I'll start the stream back up with Phys in about 5 or so. Cheers, guys.
Yeah. paper to the notes from that day.